cell division, or mitosis, I'd like you to ponder something. How does a little elephant grow up to be a big elephant? Or why is it necessary for some animals and some insects to shed their skins? The answer to that question is going to be cell division, or mitosis, and we can also call it asexual reproduction because it's one thing producing another thing. There's no fusion of anything going on here. So there are 3 reasons why cells will reproduce by asexual reproduction or go through the cell cycle. 1 is growth, 2 is repair, and 3 is replacement. So why does a cell divide instead of continually growing and getting bigger and bigger and bigger? Well, if it got too big, the cell's DNA would no longer be able to serve the needs of the growing cell. The cell has more trouble moving the nutrients and waste across the cell membrane. And because of that, as the cell gets larger, they have a smaller ratio of surface area to volume. A good example of a metaphor that we could use for this would be a library. If the cell is a city, then the DNA would be the library where we hold all the books and the information. The bigger the city gets, the more demands that are going to be on the library, more people to check out books, more books that are going to be needed, so on and so forth. And the reverse is true, or the same is true for our DNA. The bigger the cell gets, the more demands there are for copies of that DNA. And as cells get bigger and bigger, those demands get higher and higher, and it gets harder for the cell to compensate. And the same is true for all the other products of the cell. So cell division is going to be where we divide the cell into 2 new daughter cells. Before this will happen, the cell will actually replicate or copy all of its DNA so they can give a copy, one complete copy, to each of the daughter cells. Now there are 2 types of cells. We have prokaryotic and eukaryotic, and cell division is different in the 2 of them. In prokaryotes, it's just a simple matter of separating the contents into the new cell parts. We call this binary fission. In eukaryotes, they're going to have 2 stages. We have mitosis, which is what this video is about, and then we also have cytokinesis. Mitosis is going to be the division of the cell nucleus. Cytokinesis is the division of the cytoplasm. So asexual reproduction. Reproduction by mitosis is classified as asexual because they are genetically identical to the parent cell. They're clones or copies of it. This is going to be reproduction without the fusion of gametes, so we don't have 2 parents. Examples would include single-celled organisms that reproduce by binary fission or budding, or the source of new cells when multicellular organisms grow and develop. So here we have a prokaryotic cell where we copy the DNA, we divide the cell, and then we have 2 identical haploid cells that have a copy of the DNA. Chromosomes is where all the genetic information will be held. In eukaryotic cells, this is the information that is passed on from one generation to the next, and every organism will have a specific number of chromosomes. For instance, fruit flies have 8, humans have 46, carrots have 18. A chromosome is not going to be visible in most cells except for during cell division, and DNA and protein molecules that make up the chromosome spread out throughout the nucleus, so they unwind. At the beginning of cell divisions, we're going to condense the chromosomes to make it easier to divide the information. So, each chromosome is going to consist of 2 identical sister chromatids. When the cell divides, the sister chromatids separate from each other. One chromatid will go into each of the 2 new cells. And here we have a picture of a chromosome. Chromosomes look like a little X. The chromatid would be 1/2 of the X. So each pair of chromatids is attached at an area called the centromere, and this is usually located near the middle of the chromatids, or although some can be near the ends. And it's going to look like this. There will be a centromere in the center holding these 2 pieces together. So the cell cycle. The cell cycle is a series of events that the cell will go through as it grows and divides. The cells will grow and prepare for the cell division. It will divide to form 2 different daughter cells, and it begins the cycle again. One part of the cell cycle is going to be interphase, and this is where the cell will spend most of its life. It will be the period of the cell's life between the cell divisions, and it consists of gap 1, synthesis, and gap 2. Gap 1 is going to be where the cell is just doing its normal thing. The S phase, synthesis phase, is going to be where we replicate DNA. And gap 2 is where it's preparing for mitosis. And then mitosis is going to be the smallest part of the phase. We have the M phase, which is mitosis and cytokinesis. We have the S phase, which is where chromosome replication will take place. We have the G1 and G2 phase, which is the period of intense growth and activity that occurs between the M and S phases. And here again is a picture of that. So, the 4 phases of mitosis, and we're going to focus on just that little sliver of the pie. So the first one is prophase, then we have metaphase, anaphase, telophase, and then we follow it by cytokinesis. So most of the cell's life is spent in interphase. A small portion of it is going to be spent in mitosis. So about 90% of the cell's life will be spent in gap 1S and gap 2. 10% will be spent in the mitosis stage, or the cell division. And this will vary for different organisms. It can be very quick or it can be very slow. In humans it takes about 24 hours. In fruit flies it can take up to 360 minutes. So it will vary from organism to organism. So interphase is going to be the phase before mitosis will begin. The cell will look something like this. The chromosomes are replicated. The number doubles. The chromosomes appear as thread-like coils, which we call chromatin, but by the end of this phase, you're going to have the chromosomes and their copies that have been made, which we call sister chromosomes, change into sister chromatids, which is going to be where they condense by the end of the phase. So this is what interphase will look like in an animal cell or in a plant cell. The animal cell, you notice that we don't have the cell wall, but we can't really see the chromosomes yet. Prophase is the first step in mitosis. We can start to see the sister chromatids, the centrioles have started to form, and we have spindle fibers that are starting to form as well. So this is where the cell begins to divide. The centrioles or poles appear and begin to move to the opposite ends of the cell, and the spindle fibers will form between the poles. Prophase is going to be the first and longest phase of mitosis. The chromosomes become visible, and the centrioles are going to start separating and taking up positions at opposite ends. And this is what it looks like in an actual cell. You can sort of see the different chromosomes. They haven't really condensed yet. The nucleus is still intact, but they've started to condense, and eventually the nuclear envelope will start to break down. They are going to attach themselves to the spindle, which is going to attach to the centromere. And towards the end of prophase, the chromosomes coil tightly, the nucleolus will disappear, and the nuclear envelope will break down. And this is what prophase will look like in a animal and plant cell. As you can see, it's more clear in the plant cell, but you can see the different chromosomes that have started to form. Metaphase is the second phase. The chromosomes will line up against the center of the cell. Microtubules will connect to the centromere of each of the chromosomes to the two poles of the spindle. In metaphase middle, as you can see in the picture, they have lined up in the middle of the cell. So the chromatids attach to the spindle fibers, and they're going to line up in the center of the cell, kind of like this. Then we move into anaphase, and anaphase is where they're going to move apart. Apart is the third step. The chromatids, or pairs of chromosomes, are going to separate and begin to move to the opposite ends of the cell. They're going to pull them apart. They're going to look kind of like little V's being pulled apart, and it's just attached to one half of the centromeres. So the centromeres will split, allowing the sister chromatids to separate and become individual chromosomes. The chromosomes continue to move until they have separated into two groups near the poles, and it ends when the chromosomes stop moving. As you can see here, they're starting to spread apart, and this is what another picture of it, where it's separating. Telophase or telophase is where we start to see the nuclear envelope forming. Two new nuclei will form. The chromosomes will appear as chromatin. They will start to uncoil as threads rather than rods. And mitosis will end. This is the official end of mitosis. So the chromosomes are going to uncoil, nuclear envelope reforms, the spindles begin to break apart, and the nucleolus becomes visible in each of the daughter nucleuses. And you start to see where two distinct different cells will be forming. And this is how it would look in an animal and a plant cell. Cytokinesis will occur after mitosis. And this is where the cell membrane moves inward to create two daughter cells, each with its own identical chromosomes. In an animal cell, This is going to be occurring at the same time as telophase in both plant and animal cells, and it's going to be the division of the cytoplasm itself. So you're going to have just splitting of the cytoplasm. In an animal cell, we're going to form a cleavage furrow, which is where it's just going to pinch it in. In a plant cell, we're going to be forming a new cell plate in between the two new cells to form a new cell wall. However, this process can go awry. And what happens when the cells don't regulate themselves like they're supposed to is we have cell division that goes unchecked. And cancer is a disorder in which we lose the ability to control cell growth. Cancer cells will divide uncontrollably and form masses of cells that we call tumors. But what happens is you get a mutation in one of the cells, say from UV light, and after that mutation happens, it may take out the protein that helps with checking cell division. When a normal cell gets damaged, typically it will kill itself. It will go through apoptosis, and it will kill itself. It will die. In cancer cells, they don't have that signal anymore. So they will keep dividing and dividing and dividing until they form these masses that we can no longer control. And they can even get to a point where they can metastasize or move throughout the body and attack other cells within the body. It disrupts the normal activities of the individual that this affects, and it can also cause serious medical problems or even death. And it all has to do with not being able to control the cell division, not being able to control mitosis. Alright, so here are some actual pictures of the different stages. So we have interphase, which is where we have the cell nucleus. Then we have prophase, where it starts to dissolve and you have the chromatids and chromosomes starting to form. Metaphase, where it lines up in the middle, and we have the spindle fibers forming and the centrioles that are there to start to attach to them. Anaphase, where we're pulling them apart. Telophase, where we're starting to pinch it off and form the two new cells. The nuclei are starting to form. And then we have cytokinesis and the start of interphase again, where we have two new daughter cells. This is what it looks like in plants. We have interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Same process, just looks a little different. Alright, so in the cell cycle, the first part is interphase, then we have prophase, then we have metaphase, then we have anaphase, telophase, and cytokinesis. Mitosis does not start until prophase, and it ends telophase. Alright, one more time. Alright. Remember to take your notes, do your summary, and ask your questions, so don't forget to whisk.